Arnold Seawall here, BD with Panome Bio, and I'm very excited to be sitting down with Panome Bio's Chief Scientific Officer, Gary Patti, to ask a few questions around metabolomics, proteomics, multi-omics, and precision medicine. In the realm of proteomics, can you provide your perspective on the value of an LCMS approach?
Yeah, sure. So proteomics is, by definition, is the study of a lot of different protein levels. So we're talking about, in theory, the entire proteome. As many proteins as we can. And historically, we've always thought about doing that with mass spectrometry, specifically, as you mentioned, LCMS, liquid chromatography mass spectrometry. Dating back to the 90s, that was the way in which proteomics experiments were imagined. More recently, in the last handful of years, there have been some other technologies that have emerged onto the market to perform mass spectrometry, or to perform proteomics independent of mass spectrometry. And those technologies involve aptamers and antibodies, which we collectively refer to as affinity-based proteomics. It's quite interesting. Instead of using a mass spectrometer to detect proteins, essentially what they're doing is they're taking advantage of a protein having some sort of affinity to an aptamer or an antibody, and then using some sort of alternative mechanism for readout, whether it be a genome sequencer or some sort of other detector. These technologies, they're great for a lot of different reasons. It's actually extended proteomics to all kinds of different scientists, what's been my experience. Researchers who haven't been involved in proteomics or haven't thought too much about proteomics have stumbled across these technologies and are deploying them. The advantage is that they're much more straightforward to use, because mass spectrometers are complicated instruments, and the way in which these experiments are done it turns out to be a little more experimentally simple. And there are a couple of there's different organizations, different contract research organizations that offer these approaches, and so they've made it very accessible for people. And actually, it's fostered a lot of kind of a renaissance in proteomics. A lot of people doing proteomics analyses who maybe weren't before. So those are all the really great things. And overall, I'm really happy and pleased to see these developments over the last few years. That said, your question is how do these technologies compare to mass spectrometry? And I would say that the mass spectrometer is still going to provide your gold standard way of doing proteomics. One of the challenges you can imagine when you're using affinity-based approaches, whether you're using aptamers or antibodies or any other type of reagent, is the specificity. How well does your antibody or your aptamer actually bind to one protein specifically? Are you actually pulling down just the protein of interest and measuring its abundance, or are you pulling down other proteins? And that actually is fairly difficult to prove. And there's been debates in the research community about that with the aptamer and the affinity the aptamer and the antibody technologies. So there's been some controversy about the ways in which how specific it is. The other problem that you have is when you look at proteins, there's a lot of different versions of proteins. We refer to those as proteoforms. So you might have the same protein, maybe lactate dehydrogenase, let's say LDH, but it can have all different sorts of decorations on it, things called post-translational modifications. These are covalent modifications that you actually attach different kinds of chemical groups to it. And that in many cases can change the functionality of the protein. And when we look at how biochemistry is regulated, in many instances, biochemistry is not regulated by the protein levels, although that can be the case, but it's regulated by post-translational modification. So you can you could phosphorylate or dephosphorylate, and it's that process that really controls activity. Unfortunately, with the affinity-based approaches, they don't have the capacity to read out these sorts of decorative motifs. You can't measure post-translational modifications readily on these affinity-based approaches because the affinity, in most cases, isn't happening at the sites of where these covalent modifications are occurring. So they are relatively blind to proteoforms. And, I think proteoforms, we're learning more and more about proteoforms every day. And one thing is clear, that there is a lot of biology that's regulated by the proteoform. So one advantage that the LCMS technology has is the ability, because they do change the mass, and a mass spectrometer measures the mass spectrometers are able to resolve those proteoforms. So I think that's really a major advantage that opens up discovery potential and allows you to really just see beyond the simple overall average concentration of a protein, but look at all these different forms of proteins and try to get insight in what they're doing biochemically.
Well, that's great. So one approach may not be sufficient.
Yeah. The other aspect of it, to build on what your comment there, is that the affinity approaches, although the libraries have become extensive, they still are, at the end of the day, a targeted approach. I mean, you have to design an antibody, and it has to go after that particular protein that it's designed to target. Whereas when you're doing mass spectrometry, any protein that you want to look at, it's got a different mass, and you can measure it. You don't have to. There's no bias that you have to you can only look at things that are embedded in your library. It's much more global in that sense, and that works particularly well if you're doing model organism research. If you're working with zebrafish or mice or exotic plants or things like that, where in particular there's a lot of proteins that may not have antibodies. The assays have primarily been developed to look at proteins in human plasma or serum. And so that's another important point that you raise. Yeah.
Okay, great. So building on that, can you provide an example on how taking a multi-omic approach led to a discovery that would not have been possible if only one? Okay, great. So building on that, can you provide an example on how taking a multi-omic approach led to a discovery that would not have been possible if only one omic layer of measurements were assayed?
Yeah. So multi-omics there are going to be cases where, just as you say, where if you don't use a multi-omics approach, you're going to miss stuff. And whenever I think about multi-omics, I like to think of this analogy. And I'll draw this analogy with the NFL experience. A lot of, at least people in the United States, the American football, as it's called. And if you think about the NFL, I'm not giving a presentation today, but I've got this presentation deck that I like to show. If you're thinking about an NFL game and there's a very close play, did a running back cross over the 10-yard line to get the first down? Did he cross through the marker? And if you're just looking at it from one angle, you've got one camera, maybe it's the camera from the sideline. You can't tell there was a body there. I don't know. But NFL games are recorded from multiple cameras. They have like 12 cameras, an aerial camera and all sorts of cameras. And whenever they are trying to decide if you got the first down, they slow it down, and you're looking at it from every angle. And it's usually not just the one angle that they show on live TV that gives you the insight. It's looking in from that aerial angle or some other angle that does it. And I think that is the way to think about multi-omics, is that it's like looking at a football play, and each camera is a different multi-ohm layer. One's proteomics, lipidomics, metabolomics. It's a different camera angle. And so the more angles you have, the better you're going to understand what happened on that play. And when there's really close, nebulous, ambiguous things, you need those multiple camera angles to sort out what happened. And people often ask me, what's the best ohm? And I always say, it depends. It's context dependent. Just like if there's a play in football, there might be one play that camera 12 gave you that I could see it clear as day from camera 12, but all the other 11 cameras couldn't tell. And in that case, it'd be camera 12. But maybe another play on that same game, 15 minutes later, maybe it was camera 4 that gave you the superior view. So that's very high level. Let me just provide maybe a little more molecular biochemical example that people maybe can sink their teeth into a little more than that sort of philosophical analogy that I've just drawn. And we'll just use 2 ohms to make it kind of simple, and that is metabolites and proteins. So if you're doing metabolomics and proteomics. And one of the examples that I really like to refer to is the intermediates between in glycolysis between glucose and pyruvate. So if you look at all of the intermediates in glycolysis between glucose and pyruvate, and there are a handful of them, of course that's going from glucose to pyruvate. Glycolysis is that taking glucose, breaking it into two 3-carbon units, and ultimately getting 2 pyruvates. But you can also run that process in the opposite direction, called gluconeogenesis, where you go from 2 3-carbon units and you reassemble glucose. And those pathways happen interchangeably. Most of our cells are doing glycolysis, but there are, for example, if you haven't eaten yet, we're approaching the feeding time here. If you haven't eaten yet, maybe your body, your liver is doing gluconeogenesis to keep your glucose levels steady, so you can pay attention to what I'm saying until you can put some food in your body. So both those processes can be happening. And if you look at glycolysis and gluconeogenesis, every intermediate in the pathway is exactly the same. Every metabolite is the same. So if there's really no way to resolve those 2 pathways on the basis of metabolites alone, because the metabolites are identical in glycolysis and gluconeogenesis, but the proteins are not. The intermediates are the same, but if you're going in the forward direction, you use one enzyme, and if you're going in the backwards direction, you use a different enzyme. And so by looking at whether or not those enzymes are activated using proteomics technologies, that can help you discriminate which direction that pathway is actually going. So that's one example. It's just sort of a toy example of how that might provide insight. But getting back to the NFL case, the metabolites alone, it gives you you can't tell the difference because you just don't have a good angle. The proteomics in that case could actually differentiate by looking at it from aerial or from some other different perspective.
So what are your thoughts on precision medicine? How have you seen it evolve over the last decade or so? And has it actually led to better treatment outcomes? And if so, maybe how? And in my I guess limited view, maybe it seems like really only genomic testing is really considered part of precision medicine testing, specifically around cancer indications. And so why is this, and what's the limitation of that approach? And then should this type of testing really be expanded to other disease areas? And how do you see metabolomics fitting into this paradigm?
Yeah, a lot there. I could probably talk about that for the rest of the day. Let's see if we can try to tackle some important pieces there. So many great questions and a lot of really critical points. So first, let's tackle precision medicine and kind of how we think about precision medicine. And you're absolutely right. Thus far, genetics, if we think about what mutation does your cancer have, and how does that inform what therapies we use? Different mutations have better successes of being treated with different types of drugs, or different types of therapeutic approaches. So absolutely, that's a good example of how this sort of approach has already been used. But what do we mean by precision medicine? And the idea of precision medicine is that we're all different. You and I are different. And because we're different, we ought to be treated differently. The therapies that are prescribed to you ought to be different than the therapies prescribed to me. The very crude instance that I like to start with when we talk about precision medicine is if you've ever had to take, you got a headache and you take Excedrin. And you go to the back of the bottle, of the Excedrin bottle, and it says, if you're an adult, take 2 tablets every 4 hours. But there's so many different shapes and sizes of humans. Or you could imagine if you're 6'8" and you weigh 400 pounds, 2 pills of Excedrin is a lot different than if you're, a 90-pound woman. Those are very different people. And the idea that you would both take the same dosage, I think even for someone who doesn't think deeply about this, you could see immediately that from a pharmacokinetic perspective maybe doesn't make sense. So the idea is, can we learn something more about the individual, and we can use that to inform how maybe you should take 3 pills and you should only take 1 sort of situation. Now, how do we advance precision medicine? Well, the basis of precision medicine is individuality, that uniqueness that people have. And the only really way that you can get at that is by looking at lots and lots and lots of people. If you look at small groups of people, even if they're you say, well, they're representative. I'm going to look at one person from this group and one person from that group that's sick or not sick. It really does a disservice to that particular, let's just say a disease phenotype. If you say, oh, I'm going to look at 5 people who have diabetes, or I'm going to look at 5 people who are obese, or 5 people who have cancer. There's so many different shades of that condition, of that disease, that you just can't capture them all with that limited number of people. You need extremely large cohorts. Francis Collins, the former director of the NIH, was a really big advocate of this, is that the best way to change medicine by 2030, as he used to say, is by implementing exceptionally large cohorts when we do studies. So we can try to capture all of these other dimensionalities that you just can't get if you're doing a 10 versus 10 study. So moving to large cohorts, and we've seen that, the UK Biobank is a great example, where you've got extremely large groups of people who are participating in these cohorts. And by analyzing them, we're seeing all sorts of sub-stratifications and subgroups of people that we just couldn't extract. We wouldn't have statistical power if we were looking at smaller sizes. So the question is then, what is propelling precision medicine, and how can it be successful? And I think what really is key then is having technologies that can be applied to extremely large cohorts. Genomics, as you point out, that we could do that. You can sequence tens of thousands of people in a project, hundreds of thousands of people without any problem. The technologies are robust and rigorous, and that's completely, and that's why genomics has led the OMS in terms of doing that precision medicine. But can we do that for the other OMS, metabolomics, exposomics, proteomics? And the answer is yes. But it was only recently that became possible. Mass spectrometers are used at large scales. Most people are familiar that, for example, every baby born in the United States, most babies born in the United States undergo newborn screening, where they test for certain types of metabolites to determine if they have inborn errors of metabolism. And that's all done by mass spectrometry. So mass spectrometry is certainly scalable to large numbers of people, but it's only been recently that we've developed strategies to do completely global, unbiased, untargeted types of analyses with mass spectrometry at the same scale. And I think that's been a major catalyst in my mind. You asked what's the spark that's going to catalyze precision medicine, and how do you see it advancing, and why do you see it advancing?